Welcome back to the Deep Dive. Our mission, as always, is to take some really complex, cutting-edge research. Sources covering everything from mouse models to huge human studies.
Yeah, really dense stuff.
Exactly. And boil it all down into the most, well, surprising and actionable insights we can find for you, our listener.
And today, we're weaving together three pretty big areas. At first glance, maybe they seem separate, but they're actually deeply connected.
Right. What do we got?
Okay, first up, a really startling finding, how a common illness, like a respiratory infection, can apparently reawaken dormant cancer cells.
Wow. Okay.
Second, we're diving into the ultra-processed food debate. There's new evidence, including from randomized trials, suggesting UPFs are problematic not just for their nutrients, but their actual structure.
And linked specifically to things like type 2 diabetes and colorectal cancer, right?
Precisely. And then third, we look at a potential solution, or at least a powerful intervention, hands-on culinary workshops. Turns out they're remarkably effective.
Okay, so we're connecting infection, inflammation, chronic diet issues, and a practical way forward.
It's the goal.
By the end of this, you should have a totally new way of thinking about how short-term illnesses and your daily food choices might interact over the long haul. Let's jump into that first topic. It's genuinely sobering.
It is, yeah.
So the big question from the sources here is pretty alarming. Can something as common as the flu or SARS-CoV-2, can that actually affect cancer survivors maybe years after remission?
Like flipping a switch on a chronic disease you thought was gone.
Exactly. Is that possible?
Well, based on this initial research we looked at, the answer seems to be a definite yes. It's pretty profound, actually.
Where's this evidence coming from?
So this comes from a really carefully designed study using genetically engineered mice. The researchers set it up so these mice developed breast tumors, which then seeded dormant cancer cells, specifically in their lungs.
Okay, lungs are a common site for Mets, right?
Very common. And crucially, they let the main tumors regress, so these lung cells were genuinely dormant, like hidden landmines, basically.
And then they introduced the infection.
That's exactly what they did. They infected these mice with either SARS-CoV-2 or a standard influenza virus.
And what happened?
Well, it was fast. Within days, those dormant cancer cells in the lungs they kicked into high gear. Rapid proliferation, forming actual metastatic lesions. The researchers described seeing them just multiply massively in a really short time.
Okay, let's unpack this. So, was it the virus itself, the pathogen getting into the dormant cell and messing with it directly?
That's what you might think initially.
Yeah. Or was it more like collateral damage from the body's big immune response?
It was the collateral damage, and this is like the crucial distinction here. The mechanism wasn't the virus particles themselves.
Yeah.
It was traced back to a specific immune molecule that gets released when your body fights infection. It's called interleukin-6 or IL-6.
IL-6, I've heard of that. It's involved in inflammation, right?
Yeah. Exactly. It's like the body's alarm bell. It revs up the immune system to fight the threat. But it seems it also inadvertently acts as the awakener for those hidden cancer cells.
Oh, wow. So the immune system just doing its job, fighting off the flu.
Or COVID.
Right. It accidentally sends the perfect signal for those dormant cells to reactivate. That's a terrible irony.
It gets even more complicated, and frankly, the researchers use the word shocking.
Okay.
They found a follow-up mechanism. So once IL-6 woke the cancer cells up, another type of immune cell, a specific kind of helper T cell, actually moved in and essentially shielded the newly awakened cancer cells.
Shielded them. From what?
From the rest of the immune system. It basically stopped other immune cells from coming in and clearing them out. The body's own defense was sort of hijacked, perverted even, to protect the threat.
Wait, hang on. That seems totally backwards.
Yeah.
A helper T cell. Helping the cancer. Why would it do that?
Yeah, it sounds counterintuitive. The thinking is it might be a case of like mistaken identity in that highly inflamed environment. Or maybe the T cell is trying to suppress some of the inflammation locally.
And the cancer cell takes advantage.
Exactly. It manipulates that process, puts up a kind of do not disturb sign, and buys itself time to get established.
Wow. Okay, so even if those cells become dormant again later, after the infection clears, is the person sort of back to square one?
According to the sources, no. Not really. The analogy they use is really powerful. It's like trying to start a fire multiple times in dry wood.
Okay.
You might put out the little flare up each time, but you're left with, and this is their phrase, 100 times the embers you started with.
Ah, I see.
So even if that immediate metastasis is controlled, the sheer number of potentially reactivated, maybe more aggressive cells means the risk of a future relapse goes way up.
That really changes the calculation for cancer survivors dealing with everyday infections. Makes those choices feel very high stakes.
Absolutely. And look, this isn't just some weird thing happening in mice.
It's backed up by human data.
It seems to be. They pointed to data from huge repositories, like the UK Biobank, and it showed that the increased risk of cancer-related death for people who had had COVID-19 was most pronounced in the months right after the infection.
When that inflammatory response, that IL-6 surge, would have been highest.
Exactly, when things were still settling down.
So what's the clear takeaway for you, the listener, especially if you or someone you know has a history of cancer?
It's about minimizing that risk of acute inflammation where you can. The scientists' recommendation is pretty clear. Survivors should take extra precautions against respiratory infections.
Which means?
Well, that includes strongly considering vaccinations for things like SARS-CoV-2 and the flu. It's really about protecting yourself from that intense immune response just as much as from the virus itself.
Okay, that's a really important point. And it actually leads us quite nicely from that acute, maybe short-term threat to the chronic, constant threat we face daily, our food. Let's pivot to ultra-processed foods, UPFs. Specifically, the Novier Group 4 classification. Remind us what those are.
Right. Novier Group 4, these are basically industrial formulations. They're made mostly from substances extracted from foods, think sugars, starches, fats, but then combined with additives, industrial ingredients, flavors, colors.
Stuff you wouldn't find in a home kitchen.
Pretty much. And a key phrase the researchers use is that they're often engineered to be overconsumed.
Engineered. And how common are they?
It's kind of staggering. In places like the US and the UK, they make up something like 60% of daily calorie intake.
60%. 6 out of every 10 calories. That's huge.
It really is.
And we've got new data. You mentioned prospective cohort studies, really escalating this debate, linking high UPF intake specifically to colorectal cancer, CRC.
Yes, the evidence is definitely mounting, getting harder to dismiss. We looked at a big Norwegian study, over 77,000 women followed over time, and they found compared to women eating the least UPFs, those eating the most had a statistically significant increase in their risk of total CRC.
Was there anything particularly striking in that finding, a detail that stood out?
Yeah, actually, the location. The risk wasn't just general. It was significantly associated with a really striking 58% increased risk of right-sided colon cancer.
58%? Wow, just on the right side.
Exactly. And that specificity, that's interesting, right? It suggests something specific about how these foods are maybe processed or metabolized deeper in the digestive tract.
And what's fascinating, the sources mentioned this link held up even when they adjusted for things like processed meat intake, which we know is a risk factor.
Correct, which implies it's not just about the obvious bad ingredients like salt or fat in processed meats.
So it's pointing towards something else.
Yeah, the conclusion leans towards mechanisms beyond just the poor nutrient profile. They're talking about maybe non-nutrient components, additives, emulsifiers, maybe even molecules leaching from the packaging during all that heavy processing.
So it's a structural issue. Or a chemical one beyond basic nutrients.
It seems like it. It's a structural and compositional problem that our bodies maybe just aren't evolved to handle in such large amounts. They also noted the risk was even stronger in women who already had a high BMI or low fiber intake.
So those factors compound the risk.
Seems that way, yeah. They interact.
Now, the classic pushback against the whole UPF argument has always been, well, it's just calories, right? Or it's confounding factors like income or education. People who eat UPFs just have worse diets overall.
That's definitely been a major critique.
But now we have a new randomized control trial in RCT from the UK that seems to tackle that head on.
This RCT is really, really important. Crucial piece of evidence. They took participants and put them on 2 different diets for a period. One was a minimally processed food diet, an MPF diet.
Okay, whole foods, basically.
Right. The other was a UPF diet, heavy on the ultra-processed stuff. But here's the key bit.
What?
They matched both diets. Precisely. For all the main nutrients, protein, fat, carbs, fiber, even sodium and total sugar.
So on paper, the nutrition labels would look pretty much identical. Same macros, same sugar, same salt.
Exactly. Same label, just different levels of industrial processing.
And what happened when people ate these matched diets?
Despite the identical nutrient profiles, the results were stark. Participants on the minimally processed diet lost more than twice as much weight, on average about 1.84 kilos compared to only 0.88 kilos on the UPF diet.
Whoa. Twice the weight loss, even though the calories and nutrients were matched. How? They must have eaten less on the MPF diet.
They did, spontaneously. They weren't told to restrict calories, but the MPF group naturally ate less and consequently lost more weight.
Okay, this is where it gets really interesting. Why? If the macros were the same, what was driving that difference in consumption? What's the mechanism?
The main theories center on the food's physical structure and its energy density.
Meaning?
Well, UPFs are often very soft, very easy to chew, and pack a lot of calories into a small volume that's high energy density.
Right, you can eat them really fast.
Exactly. That textural change means you consume them much quicker. You swallow more calories in less time compared to, say, chewing through a crunchy apple or a steak.
It's almost like the factory has done some of the digestion for you.
That's a good way to put it. And when you eat these soft, energy-dense foods so quickly, your body kind of misses the usual cues that tell your brain you're getting full.
What kind of cues?
Things like the time spent chewing, the physical stretching of your stomach. Those signals are delayed. This is called delayed satiety.
Delayed satiety.
Yeah. Your stomach might be technically full of calories, but your brain hasn't registered it yet, so you keep eating. It promotes taking in significantly more energy before that full signal finally kicks in.
So UPFs essentially bypass our natural appetite control systems because of their physical form.
That's the argument. They're setting us up to overconsume almost regardless of willpower just because of their structure.
And we also saw new data linking UPFs specifically to type 2 diabetes risk, right, from large Korean cohorts.
We did. That reinforces the chronic impact. Looking at over 72,000 adults, they found the folks eating the most UPFs, the top quarter, had a significantly increased risk of developing TTD.
Okay.
But maybe even more powerful was a meta-analysis they discussed, pooling data from 17 different prospective cohort studies.
And what did that find?
It found a monotonic dose response.
Okay. Translate monotonic dose response for us regular folks.
Right. It basically means the risk wasn't random. It increased in a straight line. The more UPF people ate, the higher their risk.
Like a direct correlation.
Exactly. For every 10% increase in the proportion of UPFs in someone's diet, measured by weight, there was about a 10% higher risk of developing type 2 diabetes.
Wow, a straight linear increase. That's pretty damning.
It's a very clear pattern. The risk just keeps climbing the more of the stuff you consume.
Were there specific types of UPFs that seemed particularly bad for T2D risk in these studies?
Yeah, they did identify some high-risk subgroups. Processed meats were up there, unsurprisingly. Also, high-fat dairy and non-dairy desserts, think many commercial ice cream sweet puddings.
Okay.
And also ready-to-eat or ready-to-heat mixed dishes.
Yep.
In the Korean cohorts, this included things like instant noodles, which are a big dietary staple for some. Those categories consistently showed the strongest link.
Okay, let's try and synthesize this UPF section. We have to acknowledge the criticisms, right? The Novier classification can seem a bit broad or subjective.
True. Lumping a packaged salad kit in with, say, cheese puffs feels weird sometimes.
Right, and the confounding factors argument isn't entirely baseless. But looking at the weight of this evidence, especially that RCT showing effects independent of nutrients,
Yeah.
what's the big message here?
I think the overarching message that's emerging is that processing itself matters. It's a variable that has health consequences independent of just the grams of fat, sugar, or salt listed on the label.
So how it's made is as important as what's in it.
Potentially, yes. Whether it's additives messing with our gut microbes or that physical structure messing with satiety, the industrial modification seems to create unique hazards. The focus really has to shift beyond just reading the basic nutrient panel.
Which brings us perfectly to our final section, the intervention. If these highly processed, maybe structurally compromised foods are a big part of the problem, how do people realistically get the skills and confidence to move towards cooking more with fresh, minimally processed ingredients? And the answer, according to this big review paper we looked at, seems to be culinary workshops.
Yeah, and this isn't just a, feel-good idea. This was a scoping review pulling together results from 30 different studies on these workshops.
30 studies, okay.
And they confirmed these workshops offer a really hands-on, participatory way to learn about cooking with fresh ingredients. But crucially, it's not just about following recipes.
What else do they do?
They focus on building actual practical skills, boosting what they call culinary self-efficacy, basically, your confidence in the kitchen, and also sharpening critical thinking about where food comes from and how it's processed.
So it empowers people,
Yeah.
gives them autonomy.
Exactly. It's about competence and autonomy, not just blindly following instructions.
Okay, so what were the results? Did these workshops actually change anything measurable? What did the quantitative data from those 30 studies show?
The results were actually pretty impressive across three main areas. First, looking at dietary intake.
Did people eat better?
Over 90% of the studies reported significant improvements. People ate more fruits, vegetables, whole grains, and importantly, they reduced their intake of UPFs. So a clear shift.
Okay, that's huge. What about the second area? You mentioned autonomy.
Right, autonomy. In 81% of the studies, participants showed robust improvements in that culinary self-efficacy. They felt more confident preparing food, more able to make healthy choices. They felt more in control.
That makes sense. And the third area, the physical results.
Mm-hmm.
Did it translate to actual clinical changes?
It often did, especially for workshops that lasted a bit longer, say 6 weeks or more. Those interventions more commonly showed measurable clinical benefits.
Like what?
Reductions in weight, in BMI, in waist circumference, real physical changes. And beyond that, some studies reported specific clinical wins.
Oh, yeah.
Yeah, things like improved management of type 2 diabetes. They actually saw reductions in HbA1c levels, which measures long-term blood sugar. Also, better cholesterol profiles, lower total, and LDL cholesterol.
Wow.
And one study, which focused specifically on plant-based cooking, even found statistically significant reductions in blood pressure. So yes, these skills translated directly to better physiological health.
That's powerful. And we shouldn't ignore the soft outcomes either, right? The mental
No.
health side.
Absolutely not. Many studies mention those too. Participants often reported just feeling bitter improvements in quality of life, less sadness or anxiety, and also strengthening social connections through the shared experience of cooking and learning together.
Yeah, cooking can be really communal.
It really can. It taps into something quite fundamental. The key thing, though, for making these changes last.
What's that?
The review stressed that one-off workshops aren't usually enough. The interventions that worked best were longer, maybe 4 to 8 weeks, often with some kind of follow-up or reinforcement. That duration seems crucial for making the new skills and habits actually stick.
Right, it takes time to build real competence and change routines. Makes sense. Okay, this has been a really fascinating deep dive connecting these seemingly disparate threads.
It really has.
Let's try to boil it down to the 3 big lessons for you, the listener.
Yeah.
First, those acute infections, like the flu or COVID, they might actually reactivate chronic disease embers, especially cancer, which makes prevention like vaccination really critical for survivors.
Right. Absolutely. Lesson 2. Food processing itself really, really matters. It seems to impact our risk for things like cancer and diabetes in ways that go beyond just the nutrients on the label. That physical structure, those additives, they affect how much we eat and how our body handles it.
Independent of the basic nutrient breakdown. And third, that practical hands-on culinary education. It's not just nice to have, it's a demonstrably powerful tool. It improves diet, boosts health markers, and, crucially, gives people more autonomy over their food choices.
Which brings us right to our final provocative thought for you to chew on this week.
Okay, let's hear it.
If we accept that the physical texture, the energy density, the industrial composition of food are major drivers of overconsumption and chronic disease, then how useful are our current nutrition labels, really? The ones that just focus on fat, sugar, salt.
Good question.
If the processing and the structure are maybe even more important than that basic nutrient list for predicting health outcomes from UPFs, what should replace our current labeling system? What kind of information do we actually need to make informed choices and push back against the negative impacts of these foods? And how do we even begin to demand that change?
That's a big question. Definitely something to think about.
So we encourage you, maybe reflect a little this week on the structure of the food you're eating, not just the label, and perhaps think about flexing some of that culinary self-efficacy, even in a small way.
And that's a wrap on this episode of MedDecode. I really appreciate you joining me today. If something we talked about sparked your interest, or if there's a medical term that's been bugging you, let me know in the comments. And if you enjoyed this, be a pal and share it with a friend. We'll catch you on the next one.